Hi everyone, we are going to discuss about factors affecting enzyme activity in this part, which is one of the part of this enzymes topic. Okay, so let's see that. So enzymes are folded proteins that are globular in shape and catalyze biochemical reactions without being used up in the reaction. Due to the protein nature and this globular folded shape of enzymes, they are affected by several important environmental factors such as pH, temperature, okay, then enzyme concentration, substrate, product, and radiation energy, and then hydration. So we are going to discuss all these environmental factors or external factors which are affecting the enzyme activity in this part in detail. So let's begin with the first one, enzyme concentration, how it is going to affect the enzyme activity. The rate of biochemical reaction rises with the increase in enzyme concentration up to a point called as limiting or saturation point. Beyond this, increase in enzyme concentration has a little effect. So as we know, the enzyme is going to convert the substrate into the products. So if the enzyme concentration is going to be taken on x-axis and the reaction rate or the enzyme activity is going to be taken on y-axis, if you are going to see the reaction or enzyme activity, you are going to have a straight line like this. That is the as the enzyme concentration increases, the rate of enzyme activity or rate of reaction also increases. This is in one concern as you have a very lots of enzyme concentration. But here, if you are having a saturation of enzyme concentration, so obviously you are going to have a graph like this. So this is what, as I told you, it's the saturation point or the limiting factor. Until then, the rate of reaction continuously increases in enzyme concentration. So at a specific enzyme concentration, if substratum molecules are in relatively larger concentration, the enzyme catalyzed reaction attains the maximum speed. So this is how it is going to be when you are having the concentration of enzymes in relatively larger concentration when compared to the enzymes. Further increase of substrate concentration does not increase the rate of enzyme activity because all the enzyme active sites are going to be occupied by the substrate molecules. Okay, so then you are going to have this graph. Suppose if the enzyme concentration is increased, the rate of reaction also increases provided the concentration of substrate is relatively larger. Remember, if the concentration of the substrate is relatively larger, then if you are going on increasing the enzyme concentration, then you are going to have the enzyme activity also in a increased manner. If you are having the substratum or concentration of the substratum as a limiting factor, then obviously you are going to have the graph like this. Okay, so this is all about the enzyme concentration, how it is going to affect. Once again, I'm telling, at a specific concentration, that is enzyme concentration, if substratum molecules are in a relatively larger concentration, the enzyme catalyzed reaction attains the maximum speed. Further increase in the substrate, that is a substrate concentration. So here I didn't write the substrate concentration. Further increase in the substrate concentration does not increase the rate of reaction. You think that as it is going on, shall we add more concentration of the substrate? No, it won't show any, because all the enzyme active sites are going to be occupied by the substrates, because all and there will be no increase. So then you are going to have a graph like this. If the concentration of enzyme is increased, now if you are going on and increasing the enzyme concentration, the rate of reaction increases. When it is going to increase, provided the concentration of substratum is relatively larger. So in the concentration of substrates in a larger proportions, then you are going to have this graph. If it is going to have a limiting factor of concentration of the substratum, we are going to have like this. So that's how you can see the enzyme concentration, how it is going to affect the enzyme activity. Then moving on to the next one, concentration of substrate or substrate concentration. Obviously, the rate of enzyme activity increases with the increase in substratum. So this is how, this is the substrate concentration and rate of reaction. So obviously it is going on increasing. Okay. So until where it is going to occur? When there is a saturation between the enzymes, active sites and this substrate molecules. Further increase in substrate concentration will not increase the rate of enzyme activity because of limiting factor of enzyme concentration. So here, for example, 100 molecules of enzymes are there, and you are slowly adding 1, 2, 3, 4, 5, 6 until 100. So it is going on increasing. But after 100, there are no active sites for the substrate to occupy. Though you increase the substrate concentration, they are not going to have any increase in the reaction rate of enzyme. So this is a graph that shows the effect of substrate concentration on the rate of reaction that is catalyzed by a fixed amount of enzymes. Remember, here you are going to have the fixed amount of enzymes, whereas the back here you are going to have the enzyme concentration. So this graph is showing of enzymes concentration on the reaction rate at a constant level of substrate. So here is a constant and here is a large proportion of substratum, and going on increase in the enzyme concentration, you are going to have a continuous rate of reaction also increases. Okay, so that's all about the substrate concentration. The rate of enzyme activity increases with the increase in substratum, and the increase will continue till all the active sites of enzymes are occupied by substrate molecules. Further increase in the substratum concentration or concentration of the substratum will not increase the rate of enzyme activity. Why? Because of limiting factor of enzyme concentration, that is a fixed amount of enzymes or enzyme concentration. Okay, then moving to the third one, effect of temperature. Here the rate of enzyme catalyzed reaction is minimum at low temperature. Okay, and enzyme activity increases with the increase in incubation temperature up to 40 degrees centigrade, and then further increase in temperature will inhibit the enzyme activity due to protein denaturation. So this topic we had discussed in the properties of enzymes, where the temperature is going to affect the enzyme activity. So an enzyme is active within a narrow temperature, isn't it? So that is approximately of from 20 to 40, it is going to have the narrow temperature. The temperature at which enzymes show its highest activity is being termed as optimum temperature. So we know that the any incubation of an enzyme, that is whatever the temperature that we are maintaining, the same thing the enzymes are also possessing, because we call these enzymes as biocatalysts. So enzyme activity decreases above this optimum temperature as well as the below this optimum temperature. As a catalyst, they show an increased reactivity with temperature, but their properties or proteinaceous nature make them susceptible to thermal denaturation above the optimum temperature. So what is happening means, at the lower temperature, for example, if you're taking the beans and keep it in the refrigerator, you can bring it back when it is going to come to the normal temperature. But if you are going to keep it in the oven, you can't bring the nature of that bean into its normal property. So that's how the enzymes are going to have an optimum temperature where they are going to show the maximum activity. So here once again, effect of temperature, the rate of enzyme catalyzed reaction is minimum at low temperature, and enzyme activity goes on increases with the increase in incubation temperature up to 40. So here you can see, and this is the where you are going to see the maximum enzyme activity. So we can consider this as an optimum temperature for that concerned enzyme, and further increase. We think that okay, its enzyme activities or is increasing, and we further on going on increasing the temperature. What happens? Again, the enzyme activity is going to down, and finally the enzyme is getting denatured because of the high temperature. Okay, so this is how the temperature is going to affect the enzyme activity. And then moving the effect of pH. So each enzyme works in a limited pH range, so this also we know very well, isn't it? So most of the enzymes are active at neutral pH, and some are going to be active at acidic pH, while others are going to be active at alkaline pH. Examples of sucrase and the amylase enzymes. And here are the list of some enzymes where they are showing their optimum pH. That is, for example, pepsin is going to be more active at the pH 2 because its optimum pH is 2, and the trypsin is 8 where its optimum pH is going to be 8. So what is meant by optimum? Where it is going to show the maximum activity, that is going to be referred as optimum temperature or optimum pH. And urease, the pH is 7 and catalase is 6.8. So these are the examples where some of the enzymes are going to have their optimum pH or showing the maximum activity. Then since enzymes are going to be of proteins, pH changes affect the ionic characters of what we call as the amino group and the carboxylic groups that are present on the protein surface. And obviously, they are going to affect the catalytic nature of an enzyme. Okay, so as these enzymes are going to be the proteinaceous in nature, the pH changes affect the ionic characters of amine and carboxylic group of that protein which are present on the surface, and markedly affects the catalytic nature of an enzyme. Okay, so this is all about the effect of pH, which also affects. So slowly every enzyme is going to have its own pH range, and this effect of knowing the pH and temperatures, all these are very essential in the industrial purposes to study about and for the production of some of the compounds or substances. So here sucrase and then amylases, all these are going to know about this pH range. Okay, then coming to the next effect, that is product concentration. So product concentration, here I told you a term called as feedback inhibition. We will discuss in the enzyme inhibition what is this feedback inhibition, all these things. So here too much increase or the accumulation of the products that is finally forming may also affect the enzyme activity. Suppose for example, if you are going to produce more products, if that product is not going to be sold in the market, then the production will be stopped obviously. So in the same manner, if the enzymes are going to convert the substrates into products in a large quantity, and if the cell do not require that much, so obviously it is going to stop the enzyme activity. So how to stop this production means stopping the activity of enzyme. So that kind of the inhibition is called as feedback inhibition. So too much product concentration also stops the enzyme activity. Then coming to the effect of radiant energy. Yes, the radiant energy is going to be of UV rays, gamma rays, all those things, which are also harmful too. Okay, enzymes, since they are able to break various types of bonds and form new bonds. For example, if an enzyme is going to have the SS bond, okay, or it is going to form the SS bond instead of the sulfur hydryl bond. Okay, so that's how these radiations are changing the bonds where they are making the enzymes to be inactive in condition. Then coming to the hydration. Yes, the hydration, for maximum enzyme activity, proper hydration is required. So this for example, seeds which have low water content register minimal enzyme activity. Yes, if you take the dry seed, it won't grow unless and until you soak it in the water or you put it in the soil and you're adding on water. Why it is happening? Though the substrates around in them, but they won't grow, okay, or they won't show the enzyme activity. But on germination, however, the enzyme activity rises sharply. And why this germination is occurring? due to the absorption of water and consequently promotion of the kinetic activity of substrate molecules. So that's how this hydration is going to have the effect of enzyme activity. The best example is seed germination. You can take a practical and you can do it. You can see the difference without water and with water. Okay, so enzyme activity ceases when hydration is low, obviously. In our body also we should be hydrated because the enzymes have to be active in condition when you're having a plenty of hydrated condition in a cell. So that's all about the different factors of enzyme activity. Okay, here you can see the graphs. So this is the optimum temperature. So slowly the increase in temperature increases the activity of the enzymes, and further increase will denature the enzymes. And here is going to be the pH. So every enzyme is going to have their own optimum pH. For example, acidic is going to be the sucrase and alkaline is going to be of your amylase. Okay, and then here is the enzyme concentration and the substrate concentration. These graphs are showing the enzyme concentration and as well as the substrate concentration, and both are going to have their limiting factor, that is here at the constant level of substrate and here at the at a fixed amount of enzyme concentration. You are going to have this graph. And that's all about the factors affecting enzyme activity. Thank you.